Hello again. All right, we're going to do a quick recap about DNA replication. And do it as a little sketch. So really, I should be able to ask you to do a quick little sketch to explain. I'm sure it's there in your chapter objectives for chapter 17. To be able to do a quick sketch and explain what happens during DNA replication. So let's see, what colors do I have? All right, so let's say you have some DNA. Okay, so it's not a perfect drawing. It doesn't show them looking quite as nicely like a double helix, but you get the idea. Okay, so up here is the parent DNA, and we said there's a certain enzyme, little enzyme in here that's going to work in order to break apart the hydrogen bonds. Okay, so there is helicase. Its job is to break apart the helix. Oh, I guess we could label DNA. Let's say parent DNA. Okay, so first of all, helicase will break the hydrogen bonds. So if I said do a sketch, you'd make sure you label it, and then in keywords, so in small phrases, you could explain what happens. The first step would be helicase breaks apart the hydrogen bonds to separate the two strands of the helix, double helix. Okay, then anybody remember what the enzyme name is? Here. I'm gonna put some letters here so we can get the idea of what's happening. Okay, so the enzyme DNA polymerase would read the template strand, okay, the parent strand, and would cause the base pairing to be proper. So it reads the parent strand and causes the proper free nucleotides to align. And then it joins them together. Okay. Oh, sorry. Not a very good drawing. Where's my eraser? Oop, that's not good. A little too thick. I don't know what that was. Let's say it was bleak. Okay. So it causes them to be joined to make the other strand. Okay, so what was the enzyme name again? Let's see if I can get another color. Right. So DNA. Oh, why is it? Oh, let's see. Silly. DNA polymerase. DNA polymerase. Okay, and it's called that because it's making a polymer of DNA. Awesome. All right, so this class, we're going to start talking about, or sorry, this lesson might be in the same class as the other one. In fact, it probably is. So we're going to move on to talk about gene expression. Okay, DNA expression is another way you might hear it called. Basically, when we talk about gene expression, it means protein synthesis. Um, when we looked at this before, it was when we talked about how steroid hormones work. Basically, the hormones go in, um, travel into the nucleus with a hormone receptor, and they bind to the DNA and they turn on this process. Okay, so there are other things that can turn on this process, but one thing is a hormone. So if a steroid hormone comes in, it can cause this to happen, and then the action of the steroid hormone works through the proteins that are made. Okay, but proteins are made for many different reasons, not just in response to hormones, but also in response to other things, other factors that can turn the genes on. Maybe it's a factor during development. So a certain factor will go and say, oh, okay, the baby's at this stage now. We need to have, in this particular cell, we need to have this happen right now. So it turns on certain other genes. Okay, so if you think back to what you learned about in terms of hormone action, then it's the same kind of process, but it might just have been triggered by something else. So to express DNA, or for the gene to be expressed, it has to go through the two steps, transcription and translation. Transcription, if you remember, goes from DNA to messenger RNA. Okay, and translation, the messenger RNA is read and translated into a protein. So the amino acids are put together in order to make the protein. Question is, why code for proteins? Well, proteins are used as building materials to make things like cell membranes. If you remember last year, we drew that big cell membrane with all the phospholipids and the 6 different types of proteins, receptor proteins, transport proteins. They have lots of different jobs right in the cell membrane. They're also organelles. So if you think of all the microfilaments and microtubules, anything made with them, so flagella, cilia, any of those things, they're made up of proteins. The stuff in the membranes themselves are made of proteins, of the membranes of the organelles. Other body structures that are more obvious to us, like your hair, your nails, the pigments in your skin or in your eyes to give color, they're all proteins. Some of them have extra special jobs. So some of them are hormones that we've seen, which carry messages and help direct body functions. Some are enzymes. We have many, many, many enzymes in our bodies, because if we didn't have the enzymes to speed up the processes, then we wouldn't, our body cells just wouldn't be able to exist. The reactions would go so slow that we couldn't function. So enzymes are really important, and the membrane proteins that we talked about. Each gene can produce only one type of protein, so one enzyme or other type of protein. So the DNA must contain many different genes for the different proteins. And if you remember, the DNA that we have in our cells is the same in every cell. It's just that certain ones will be turned on in certain cells. So for instance, if you're trying to produce hair, then it's not going to be a liver cell that's going to turn those genes on. It's going to be a cell that's in an area where you want to make that particular thing. Okay? Oh, sorry. There is a link there that you could go to the tour site if you want to, or you could just have a look at it on your own time. I'll leave that up to Miss Powers' discretion. Okay? So the first process we said was transcription. And you know from when we talked about it before that it's a process in which a segment of DNA is transcribed or copied to produce a segment of RNA. And in this case, since the RNA carries the information or message coded by the DNA, we call it messenger RNA or little m RNA. Its job is very important since DNA, we said before, is so big it cannot leave the nucleus. So the messenger RNA travels to the cytoplasm where ribosomes can bind to it and read the code to produce the proteins. So first of all, from what we talked about so far, you should be able to tell me some differences between RNA and DNA. And understanding those differences is important for keeping DNA replication and transcription and translation all kind of straight. So, if you look at the names themselves, RNA, DNA, the NA stands for nucleic acid in both cases because that's the type of macromolecule they are. The first letter tells you which sugar is in it. Okay, so RNA is ribo for ribose, ribonucleic acid, ribonucleic acid. I was just trying to underline the R there. Anyway, DNA uses deoxyribose for its sugar. So one difference, a big difference, obvious from its name, is that they use a different sugar. You should be able to tell me which it uses. Okay, if you remember ribose, then the DNA is missing. It says deoxy. It's missing an oxygen compared to the one that's using RNA. Okay, so DNA stands for deoxyribonucleic acid. DNA. Awesome. RNA is single-stranded and small, right? DNA is really big and it's double-stranded. RNA uses uracil. So that's A, C, G, and U are the nucleotides. A, C, G, and U. Whereas DNA, we said, is A, C, G, and T. Okay? And because RNA is small, it's so small that it can leave the nucleus and go into the cytoplasm. DNA is so large that it can't leave the nucleus, and that's huge. Okay, we have to make a message form so that it can leave the nucleus and then cause the next process to happen out in the cytoplasm. So if you look down below, it's highlighting these features. Okay, it uses a different sugar. RNA uses ribose, DNA uses deoxyribose. RNA is single-stranded, DNA is double-stranded. Okay, I think that's about it for that one. So you should be able to tell me, it'd be a good compare and contrast practice question. How are they similar? How are they different? Okay, don't just tell me they're both nucleic acids. Okay, well, that's fine, it's in the name. That's pretty weak for an answer. If they're both nucleic acids, you could say they're both polymers of nucleotides, or they're both made up of nucleotides, for instance. There's lots of other similarities that they have. Okay? Okay. So, steps of transcription. the player's part is just saying the key things that are involved. So for transcription to happen, you need DNA, you need the nucleotides that are floating around in the nucleus, and you need the enzyme RNA polymerase. Not DNA polymerase, because this time we need to make RNA. So first step is the DNA has to be unzipped. In other words, the hydrogen bonds have to be broken in the region that's going to be transcribed. So there's going to be some initiation factors. We're not going to get into all that much detail, but basically some little chemicals are going to come and bind and kind of say, okay, here's the gene, this is where it needs to be opened, and it causes it to be opened in that region. So for our purposes, you can know basically the DNA is unzipped in the region of the gene to be transcribed. So in other words, the hydrogen bonds are going to be broken. Base pairs are separated. The enzyme RNA polymerase is going to read the template strand, and it brings, so here's the template strand here, here's the enzyme, and the enzyme is going to read the template strand and add nucleotides. So it brings the corresponding nucleotides, RNA nucleotides, A, C, G, and U, so that they bind in the proper base pairs. It joins them together to form a single strand, which separates from the DNA and can go to the cytoplasm, right? The two DNA strands are going to go back together so that they're stable again, and the messenger RNA will go out into the cytoplasm to be read by the ribosomes, in other words, to be translated. There's a link here for an animation. Hopefully the link is working. If not, it's linked directly from the blog. So if that link is for some reason, sometimes they change the address. If it's not working, then you can use the one right from the blog. Okay, so if you want to just pause this, and then you can use the one from the blog if you need to. What am I thinking? You're going to have to use the one from the blog because this is a video. I'm silly. All right. So, perhaps I should stop that there. No, we'll keep going. I'm sure if we're getting too long for a class, that you'll be able to stop wherever we get. The next part is making of the protein. So in order to understand the messenger RNA, how it's going to be translated, we have to understand what goes into making the protein and what kind of code we're looking at. So protein is made by linking the building blocks. And we know from last year, or I hope that you remember, that those are amino acids. They have to be joined in a specific order, just like beads, certain shape and color on a necklace. One change can result in a different protein with different properties and functions. So if there's a mistake, then it can really, really affect the protein. Might even make it so it doesn't get produced. So the sequence of amino acids is really important. There can be as few as 8 or as many as thousands. So it depends on the protein, its structure, what its job is, how big it is. Within the code, there are 3 letters. Okay, so basically we're trying to code for about 20 different amino acids that go into making up protein. There's only about 20 of them, but they're joined in different ways. Just like you can have in DNA, you can have A, C, C, G, U, A, like a different order of letters, only with 4 letters. Okay, somehow we've got 30,000 genes with 4 letters, but with proteins, we have about 20 different amino acids. Each of them is coded for by, like I said, 3 letters, 3 nitrogenous bases in the messenger RNA strand. This 3-letter sequence is called a codon. Think code. It has the code in it. It's a codon. There are 64 codons. So if there's only 24 amino acids, but 64 codons, we must have more than one codon for each amino acid. And that is good in some ways because it allows for silent mutations or mutations that don't really have an effect on the protein. Kind of a wobble thing. You learn more about it in university. Okay, so this is, one sec, wrong way. Okay, this is the code, and this is the exact one that you will have on your exam. So if you learn how to read this, you will have this on your exam in order to decode a piece of DNA. So the way that it's done is we have 3 letters to look for. Okay, the first letter is given by these over here. Okay, so if I was trying to find out what amino acid is coded for by UCA, okay, I would look. This is my first letter, so I'm going to go for the first row for U. These along the top tell you what the second letter is. So I want UCA. So I need to go in here. Okay, UC. And then these ones over here are the third letter. So I want UCA. It should be right here. So UCA codes for the amino acid serine. I don't expect you to know that it's serine, S-E-R-I-N-E, but I do expect that you should be able to tell me its 3-letter abbreviation, S-E-R. Okay, so I want you to tell me, or try telling me here, I'll get that stuff out of the way. Oh, maybe I won't. I need an eraser. That's not an eraser. There. All right. Okay, so I want you to tell me what amino acid is coded for it, and I want you to look at your own sheet. You could probably see it better on your own sheet. There should be a bigger version of it, maybe on the back page of your booklet. I want you to tell me what amino acid is coded for by the codon AUC. Okay, so again, here's the first letter. So I want A. U is the first column. AUC. So it would be isoleucine. Isoleucine. I-L-E. Okay, let's try C-A-C. Okay, so I know it's going to be in this row because it starts with that letter. C, A is the third column. So C, A is over here, somewhere in here. I want CAC. There's CAC. CAC codes for histamine. Okay. Again, I don't expect you to know the full names. You're not going to have to memorize this chart, but you should know how to read it. You're going to have to tell me what the amino acid sequence is for the protein that's being coded for. Okay, so you're going to have some practice at this whenever we do a problem a little bit later, but you'll also have some practice whenever we do a little mini lab, which will probably be tomorrow or later this class. All right, so that is the code. That is how it's broken down in 3 letters at a time. Okay, I'm going to pause it there. So that's how you read the message that's in the messenger RNA. I'm going to stop the video and I'll do another section for translation.